Hello everybody, once again, this is your biology channel, Dhananjai Bioguru. Today we will discuss a very important topic, the transfer RNA, the soluble RNA, its structural organization, its functional organization, all the important pointers that may ask in your NEET examination and your board examination. So please stay tuned. Let me start the transfer RNA molecule. Transfer RNA or soluble RNA. As the name of transfer RNA signifies the role of transfer RNA. What the basic function? Transfer RNA. Soluble RNA will tell you the structural organization. These two names are, one name stands for structural organization and other name is important for the functional organization. Transfer RNA is the functional structure of transfer RNA and soluble RNA is the structural organization of the RNA. Let me just simplify these two terms. And the transfer RNA also known as adapter molecule. So you know transfer RNA is called adapter RNA molecule. We also call it adapter molecule or adapter RNA molecule. So these names are very close to each other, but let me briefly discuss why we have different name of the transfer RNA. So first thing is that transfer RNA involved in the translatory process. Transfer RNA read the genetic codons present in the mRNA, and genetic codes carry the genetic information that transcribed in the form of mRNA on the DNA template. DNA of course will not come in the cytoplasm and do all the necessary biological process. Any metabolic activity, all the metabolic activity is in the control of RNA. RNA are three types: messenger RNA, transfer or soluble RNA, and ribosomal RNA are the RNA that involved in the formation of ribosomes. So now today we know about the transfer RNA. How does transfer RNA function? What is the structure of transfer RNA? What are specific properties of transfer RNA? So let me one by one discuss here. So first thing is that you got the name transfer RNA because they transfer the genetic information. They read the genetic information that is embedded in the form of genetic code in mRNA. They read it and transfer that information in the picking of a specific amino acid. So the transfer RNA has a special biological capability to pick up the specific amino acid as per as the information processed in the genetic code. So thereafter, once they read the genetic code, they transfer the information in turn to pick up the specific amino acid. Later these amino acid polymerize, they form the polypeptide bond and they form protein. So protein is the translation of genetic information. Protein is the translation of genetic information as a metabolites. Is it clear? So you remember, so there are two things are very side by side: transfer and translation. And transfer RNA perform these two dual functions. By reading the genetic code, they transfer the information, the genetic information from genetic code to picking the specific amino acid. Later that amino acid translated means they translated in the form of protein. So what is the protein? Protein is the translation of genetic information as a metabolite. So these are the two terms to remember. Why we call it transfer RNA and now why we call it soluble RNA? Because they have a very strong affinity to different enzymes, to codons in the mRNA, to ribosomes, as well as to affinity toward the specific kind of the amino acid. So why known as solubility? So solubility is due to two adapter and solubility. So if the two terms adapter and solubility, adapter and solubility are the terms that are given to transfer RNA because of these four things. Number one, strong binding with one, they bind with a specific amino acid, the transfer RNA have a strong binding end, the third end of the very soon I will explain you. So they have a strong affinity or binding affinity with the amino acid. There are 20 different types of amino acid, and these amino acids, bind with the transfer RNA as per as the genetic information transferred to the third end of the amino acid, tRNA. And number two is, look here, enzyme link. They can easily link with the enzyme linking site. And number three, ribosomal binding site. And number four, anticodon site. Anticodon end means binding and reading genetic codon on mRNA. So, simple way you can see here the transfer RNA molecule has possess a very strong binding affinity with four different, molecules, four different, event. Number one is the amino acid, number two is the enzyme, and so on. So transfer RNA have a capacity to bind with the ribosome, capacity to bind with the special kind of specific amino acid, tendency to bind with enzymes. They are very important for the translatory mechanism, and is very specific to bind with the genetic code present on the mRNA, and that end of tRNA is known as anticodon end. And next point is, how does transfer RNA synthesize? Next big question is here, how does tRNA get synthesized? All the RNA synthesized in the nucleus, mRNA synthesized on the DNA template as a transcripted language of the DNA. mRNA is nothing but the transcripted language of DNA. Because DNA, of course, in reality, do not exist outside the nucleus. A lot of enzymes that are present in the cytoplasm, they will break the phosphodiester linkage in the DNA, and DNA will destroy the cytoplasm. So DNA will not able to come in, outside the nucleus. So how does the genetic information, but all the metabolic process going on in the cytoplasm? So DNA process the genetic information through its own transcribed copy. So DNA transcribed genetic information in the form of genetic code, and the genetic code sequence is known as messenger RNA. So during the time of transcription, simultaneously transfer RNA synthesized inside the nucleus. So where does during transcription of genetic codons in mRNA, the chain of genetic code is of course known as mRNA. So during the transcription of genetic code in mRNA, what happened? Transfer RNA synthesize. Meantime, so their transfer RNA, suppose that 84 genetic code has been transcribed in the form of mRNA on DNA template. So there will be the possibility of 64 transfer RNA. 84 your transfer RNA, but that figure may be slightly fluctuate. I will tell you. So normally they are total. Total how many genetic codes are here? They are total genetic code equal to 64. So in this way, there must be 64 transfer RNA synthesized simultaneously with the transcription process on the DNA template. But you find if the exact number of genetic code 64, transfer RNA will never exceed the number of genetic code 64. It will be lesser number. Imagine if in the transcription on the transcripted chain of messenger RNA you counted 324 genetic code. So what might be the possible number of transfer RNA? Not 64, not 364. It should not be exactly matched to 324. Sorry, the number is here 324. So there should not be 324. Although the transfer RNA parallelly simultaneously synthesized in the nucleus along with the transcription of the genetic code in mRNA. So what the reason? Total nitrogen bases are 4. Vowel concept, are shown you. Total genetic code are 64 due to vowel concept. Very important. Total number of genetic code is equal to 64. Among these 64, 3 codes are nonsense. That are the inert code. Out of 64, 3 codes are nonsense. Nonsense means they are inert. 3 codes are inert. It means they are the genetic code that are not specify any amino acid. They will not specify any amino acid. So these genetic code are inert. If the particular genetic code will not codify, will not specify any amino acid. So what happened? They will not require any transfer RNA because they will not participate in the sequencing of the amino acid. They will not participate in the picking of the amino acid. So 3 codons are nonsense codons, are inert codon because these codes are inert. They do not specify any amino acid during translatory mechanism. So why 3 codes are inert? So out of 64 codons, these 3 codes do not require any transfer RNA. And these codes are just UAA, UAG and UGA. These are the 3 codons. And whenever these codons will come across the translatory process on the ribosome head, they will not pick up any transfer RNA because they have no transfer RNA, and that person translation will stop. So why these 3 codes are known as stop codons. Stop codon. So stop codons have no transfer RNA. They have no any transfer RNA. So right now, how many genetic code remaining? There are 61 genetic code remaining. So for this 61 genetic code, there is a maximum number of genetic code that will specify any amino acid. So among the 64 genetic code, there are 61 genetic code that will specify the specific particular amino acid. So there are 61 transfer RNA molecule. So now total 61 transfer RNA gets synthesized in the nucleus simultaneously with the transcriptional process. So you remember this thing, remember. So only 61 types, and among 61, one transfer RNA will be initiated. That is AUG. Okay, the one genetic code is AUG, that stands for the picking of an amino acid. Methionine is the first amino acid that will start the translatory mechanic. The first transfer RNA that will pick the amino acid methionine by reading the genetic code AUG. So the first transfer RNA that will pick the methionine is known as initiator transfer RNA. So the transfer RNA that read the genetic code AUG is known as initiator transfer RNA. So you do remember, very important, very important. So now out of 64 genetic code as per Bobble hypothesis, there are only 61 genetic code that will translate any amino acid in the form of polypeptide formation. So 61 types of transfer RNA found both in prokaryote and eukaryote, and 3 are the stop coded. So I give you the example. Suppose that on a DNA template during the transcription, 324 genetic code transcribed in the form of mRNA. In the mRNA, what will be the possible, and what will be the possibility of transfer RNA there will be? Suppose that if in the sequence I say there are 28 are nonsense codon, so you remove 324 minus. Suppose that if I say out of these 324, 21 are nonsense or inert codons, inert. Then the exact number of transfer RNA required for the translatory mechanism will be 324 minus 21 is equal to 301 transfer RNA will form among the given 61 types of transfer RNA. So the maximum types of, there are 61 types of transfer RNA, and among the 324 there are 20, then you minus 324 minus 21 nonsense genetic code. Nonsense had no transfer RNA, the number exactly equal to 301 transfer RNA, and these 301 transfer RNA will be different 61 types of transfer RNA, depends upon the genetic code that transcribed in the mRNA chain. So this is all about the origin of the transfer RNA. Now we come to structure. There are 2 types of structure have been seen for the transfer RNA. One is the primary structure. There are 2 types of structure. Primary structure, 1 degree structure, and second is a 2 degree structure known as secondary structure. Secondary structure is structural organization, important. Structural, and 1 degree is functional. When transfer RNA comes in play, means comes in its role to pick up the amino acid in the translatory mechanism, then tRNA found in the 1 degree primary structure, that is very unstable, and if the transfer RNA do not involve in the picking of amino acid, they remain in the form of 2 degree secondary structure. 1 degree structure is known as L-shaped, which is L-shaped structure, and 2 degree structure is known as clover leaf model. This structure is called clover leaf model. Why clover leaf model? You can see here, this is the, now we should start from 15. The nucleotide always starts from the 15. This is the one loop is formed here, then the second loop is formed here, then a minor loop, and then third loop, and this is the end of the tRNA polynucleotide chain. So why the tRNA is known as soluble RNA or adapter RNA? Because they have a strong affinity toward this loop bind with ribosomal binding site. Ribosomal binding site. This end is enzyme binding site. The enzymes that involved in the translatory mechanism. This end is known as anticodon end. Anticodon end. Loop are known as genetic code reading end or genetic code binding site or mRNA. So anticodon loop or anticodon site, you can also say genetic code reading site. This site is known as genetic code reading site or genetic code binding site on the mRNA, and this end is known as specific amino acid binding site. So now there are 4 different sites that present in the here in the transfer RNA. So this structure clearly tell you the tRNA is an adapter molecule. So the clover leaf model explain you, present you and clarify you the stability of the transfer RNA molecule and say it's soluble tRNA, say it soluble adapter tRNA molecule. But when you come and see the L structure, is a highly functional and unstable structure. Here, this structure is known as primary structure or L-shaped structure or functional structure of transfer RNA. Why the clover leaf structure is very stable as compared to L structure? Let me see. Due to enfolding of the chain of transfer RNA from 5'3", there is a slight hydrogen bond formed, like in DNA molecule, but these hydrogen bonds are not continuous and vigorous. So here you can see the hydrogen bonds are formed, So these are the hydrogen bonds formed, like this one. So due to formation of hydrogen bond between overlapping region, between the looping region of the tRNA molecule, they have gained extra stability. So transfer RNA molecule is now stabilized in the 2 degree or secondary structure due to enfolding of the polynucleotide chain, and there are major loops created. One loop have high enzymatic affinity, second loop have high ribosomal affinity, third loop is the anticodon end that have affinity to the genetic codon mRNA, and third end is also the specific amino acid linking site. So here you can explain the structural analog of the tRNA molecule through different positions. And now when they come in the action, they become the L shape. This bond has been break. Now they separated. Only the most prominent is anticodon. This provide the template for the attachment of codons on mRNA. They attach to the ribosomes, they attach to the enzymes, and third end will pick up the special or particular amino acid through the genetic information processed on anticodon end, and later they translated in the form of the polypeptide chain that is known as protein. So that's all about the transfer RNA. Very important and role and everything is so highly elaborated in the mechanism and in the structural aspect. So don't forget, go each and every aspect of the tRNA, either functional or structural, and one of the core topics in the molecular basis of inheritance in the gene expression, simply the three core area of the molecular biology: replication, transcription, and translation. So translation mechanism very much a control by the tRNA molecule. So in the next class I will move with the transcription process, where we shall study how does the mRNA transcribed and processed, and later the functional aspect of the mRNA. Thank you very much. Till stay tuned up, and if you have any confusion now, please write in the comment box. If you go through the video, please you subscribe the channel, and if you find a lot of information in the video, you also like the channel. Okay, thank you very much once again.